Good day, class. Thank you so much for tuning on to Metric Hub. It's all about education, nothing more, and empowering our youth, ensuring that you reach your destination the shortest possible time, no repeats. Connect to Metric Hub and you know that your things are done. Today, I'm going to be presenting to you uh, protein synthesis or translation, the process through which proteins are formed. So displayed on the board is just one other diagrammatic representation which you need not to forget and you need not to misrepresent. So I'm going to be talking and explaining everything about this diagram where you are seeing the codons, anticodon, ribosomes, and every other things that you are seeing mRNA and stuff here. So I will come back to this diagram after introducing to you the chapter or the content of today's subject. So check here. Today we are going to be talking about translation. It is the process of synthesizing proteins based on the sequence of mRNA molecule. Proteins are formed. The process whereby proteins are formed based on the sequence of mRNA molecule as it is read by ribosome is what we call protein synthesis. It can also be defined as the process during which the chemical instructions on genes are used by a living cell to make proteins. One key thing that I want to emphasize right now to you is that it's a process which during which the chemical instructions on genes are used by a living cell to make proteins. I'm sure that how do we relate to that to say chemical instructions on genes are used by living cells to make proteins? This is a process that starts from where? From transcription. So when transcription has occurred, we are saying the genetic information that is on the genes is being transferred into or transferred to the mRNA, which is going to be used during the protein formation. That is what this whole process, or that is what all this is all about. Process by which mRNA makes proteins. In simple terms, we can say that that is protein synthesis. So please take note of this definition. Please take note of the definition based on the sequence of an mRNA molecule. Chemical instructions on genes are used by living cells. Living cells, they do grow, they do repair dead cells. So when dead cells are to be repaired, when there are living cells are to grow, everything is all about protein formation. I'm sure that you remember that proteins are the building blocks of living things, which is one key area of our subject that we are supposed to explain and discuss today. So without wasting any time, let me just go quickly to the diagram because most times you see that they are at grade 12, we are given diagrams and we are supposed to know what is the diagram representing, and then we can be able to answer every other question that might follow. So in this case, what is happening is that you can see yesterday or in the previous presentation, I tried to illustrate the process of protein synthesis. So, protein transcription. So, as you can see here now, other writers or other authors, they do present the protein synthesis process as being shown here. So, what I want you to know is that this process is taking, we can name them as process A, and then we can also name this as process what? As process B, which is taking place outside this circle or outside this double line here. So one key, some few important things that I want to emphasize is that this process is taking place, it is taking place in the nucleus. Where does process A occur? It occurs in the nucleus of a cell. It occurs in the nucleus. I'm sure that you should understand that it occurs in the nucleus of a cell. That is where it is occurring, process A. And then process B, it is taking place in the cytoplasm. It takes place in the cytoplasm, which means it is outside the nucleus. The moment you see that, you can see that there are two processes that are there or that are to be explained. So process A, we explained it in the previous presentation. Process A, it is what? It is transcription that is being explained in this case. Here we are shown that this is transcription that we are to understand in process A, and then process B is therefore what we need to understand as what? As it's process B, it is now protein, process B is protein synthesis, which is also translation. So what is important for you is that you should understand that this process, sometimes other examiners or any other writers or whatever, they can present this question in this order, where you see that A, we don't want to tend to say anything about it. We don't want to explain anything about A, but B is where we are focusing on, specifically on protein synthesis. So I just want to label this part B. I want to label it. I want to label part B, and if you can see here, the arrow is showing a nuclear pore. Arrow is showing nuclear pore. This is the nuclear pore that I was referring to, or which I mentioned last time. The nuclear pore is shown by this arrow, which is showing the movement of mRNA out of the nuclear membrane. So I also mentioned to you that this is the nuclear membrane, which we also mentioned last time when we did transcription. So that is nuclear membrane, and that is the nuclear pore. And obviously, we are saying other times, just to make things clear, they say identify molecule X. Molecule X, if you are saying molecule X, what you need to know is this is a double helix, not just DNA molecule, it's double helix. It's DNA double helix structure. This is the DNA double helix structure. X is DNA double helix that you can see there. And then otherwise they would want you to see here at which we can add the label to say what is this one. We said this is mRNA. That is mRNA that is formed there. And then obviously the process taking place in this zone, the process of transcription will be occurring in this zone, which I have marked as to say that this zone which I'm shading here, it is representing the process of that is where transcription takes place, and then that occurs. So let me just quickly label this part B of our diagram. What is part B? So certain times again, we might find out that we would have things, or we would have proteins which are just around, or which are in the cytoplasm, which we can give to say that these are protein. We can say that these are, we need to know that these are what? They are free amino acids. Free amino acids, which I'm sure that you remember amino acids are the building blocks of proteins. And then I want you to see that this part, this chain, we need to know that it's a growing chain, it's a growing protein chain. This is a growing protein. It's a growing protein chain. The growing protein chain is that one, which we are seeing the dots that are joining together there. And then at certain times again, I would want you to know that I want you to know and also identify on this diagram this part here. Let me just use, I'm saying this other part here. which is shown by this arrow. The arrow is showing the 3 nucleotide bases. We are saying this is called what? These are called anticodons. It's called anticodons. That is anticodons here. This, I'm talking about these are called anticodons. As you can see, I'm talking about the 3 nucleotide bases there, which is also shown on what? On our tRNA. This is our tRNA. Our tRNA is shown there. And then we can further say that we need to label this. What is it that we are seeing here again? We also see the codons. The codons also, which are represented by that arrow showing there. So tRNA is also just the same thing as to say this is the dotted arrow here is just showing these two are the same. We are showing that those are that is tRNA, and then at another point again we need to see that this is the ribosome. That's the ribosome there. That's the ribosome that you are seeing there again. And then is our ribosome. What else? What is it that we have not labeled? The ribosome. We also have to understand and label to say that uh, this it is showing the mRNA being translated. I can also label this and say that it's mRNA being translated. I'm talking about this one. Is mRNA being translated? What else can I need to maybe want to, should I have to label there? What else should I have to label? What else should I have to label? So what is happening is that the tRNA is bringing what? It brings the amino acids to the ribosome, as you can see there. The tRNA is bringing the what? It is bringing the amino acids, which you have seen. I've labeled that we have got the amino acids, which you are seeing as these dots are the free amino acids that you are seeing, which I have just put as to say these are just the dots that are there, indicating that the free amino acids, as you see, they are attached to the what? To the tRNA that you are seeing there. So understand process A takes place in the nucleus, process B takes place in the cytoplasm. And then I mentioned to you that it is out of the nucleus, process B, protein synthesis, because it has moved through the what? The nuclear pore, and then it is there to attach itself to the what? To the ribosome that we have mentioned there. So I want us to quickly go to the next let me just go straight to the components. These things that I mean, all the points that I've labeled here, the codons, we are talking about the bases that are found, the 3 nucleotide bases, that are found on the mRNA. I can say 3 nucleotide bases. 3 nucleotides bases on mRNA is what you call the codons. Obviously, we are saying on what? 3 nucleotide bases. This is also 3 nucleotide bases on tRNA is what you call the anticodons. So I want you to look at this and understand them very clear. I'm saying here, you must not make mistakes. You need to understand the process very smart and clean. There is something unique there, which we are seeing the anticodons are 3 nucleotide bases. bases on what? On the tRNA, and the codons are the 3 nucleotide bases on what? On mRNA. And then let me just go quickly to the what? Let me just go quickly to this the components, the components that are the components that are that we can see in the process of translation. Components useful during translation. Like I'm saying, I've been mentioning these codons. What? So codons are the instructional codes used by cells to identify which amino acids are to be used to assemble proteins. You can see that they are instructional codes that are used by cells to identify which amino acids are to be used to what? To assemble proteins. So this is they are actually there to commission or to actually identify or to give instructions on the specific amino acids that are to be used in protein synthesis. So there are 3 nucleotides in length. I'm sure that I told you that I've mentioned this again, that these are 3 nucleotides in length. There are 3 nucleotides in length and are read by the cells as its machinery moves down the strand of the gene. The strand of the gene we are just talking about is the strand of the mRNA. The mRNA, the strand of the gene, or the strand of mRNA, the strand of the gene, or you can say the strand of mRNA. That is exactly one and the same thing. The strand of mRNA, the strand of the gene or strand of mRNA. Remember that the genes are contained on mRNA during the process, or they've been sent there during the process of what? Transcription. So nucleotide codons and their corresponding amino acids, nucleotide codons and their corresponding amino acids. What happens there? We said that they are to identify which amino acids are to be used in assembling of proteins. So what is happening here is that I want you to look at this to say that the nucleotide codons, and their corresponding amino acids. So here, what you need to understand is that each codon has got its own specific amino acids. I'm going to go and look into this. Check, for example, we are saying the TGC is thymine guanine cytosine. It has got it has to identify an amino acid that is called what? Cysteine. And then we have got CTG. It is corresponding, or it codes for leucine amino acid. We have got AGT. It is corresponding or coding for the serine. We have got GCA. It is coding for, we have got GCA, which codes also for alanine. So what I'm saying is that each codon has got its type of amino acid that it must meet, or it must assemble, it must identify, which must be brought in by what? By the anticodon. So having said this, let me just quickly give you another thing again that is very important, which is the difference between the codon and the anticodon. So a codon is a sequence of 3 nucleotides in DNA or RNA, a sequence of 3 nucleotides in DNA, or it can be in RNA. Well, an anticodon it's a sequence. It is a sequence of 3 nucleotides in transfer RNA. I'm sure this I have already mentioned there, to say that the 3 nucleotides that are in what? In tRNA, they are called anticodons, and then in the codons are in the what? We can find them in the DNA or in the what? In the RNA. So codons code for amino acids, which I already mentioned again, that the codons, they do code for specific amino acids, while anticodons make the anticodons, they match codons to what? To amino acids. The anticodons class, they do match them to, they match codons to amino acids. The codons code for amino acids. So what we are saying is that the codons, You've got an information, they've got a template, they've got information which must relate or which must identify specific amino acids. So the anticodons, they actually match the codon to their amino acid that they must match to. So it is the duty of the anticodons to bring the corresponding amino acid to the what, to the codons. So codons, they code, they resemble, or they've got an information that identify specific amino acids. So the amino acid that it is identifying, it is matched to it by the anticodon, which is from the tRNA, So I'm saying components that are in protein synthesis. We do have the messenger RNA, which I also mentioned last time, is a single-stranded RNA molecule that is complementary to one of the DNA strands of a gene. Remember that the DNA strand of a gene is complementary to what? To mRNA, in the sense that mRNA is formed from the template that is from the DNA. The DNA template is used to transcribe or to form the mRNA structure. I've explained that in my previous presentation, that mRNA, it is formed using DNA, using one of the strands of DNA as a template. Hence, we are saying it is a single-stranded molecule that is complementary to one of the strands of DNA of a gene. So the mRNA is it is actually an RNA version of the gene that leaves the cell nucleus and moves to the cytoplasm, which I mentioned again in my previous presentation that even here in the beginning, I said that it moves, it leaves the cell, it moves out of the cell, it leaves the cell nucleus. That is, when does it have to do that? It has to do that towards the process of protein synthesis, preparing for protein synthesis, where proteins are to be made. So it is going to go away to the cytoplasm. That is one key thing that I want you to understand. And then transfer RNA. It is actually small molecule in cells that carries amino acids to organelles called the ribosomes. I'm sure that I've also said that where they are linked to proteins, and then it functions also. The tRNA also functions as a shuttle. It functions as a medium. It functions as a shuttle, as a transport medium to get amino acid to the ribosome. It is responsible for getting the amino acids to the what, to the ribosome. That is one other thing which you need to understand with regard to the tRNA. What is the difference there? There is quite a lot of information that we cannot exhaust in this presentation, but for the sake of progress, let's go on. The ribosome is actually the cellular machinery responsible for synthesizing proteins. As you can see, we are saying the ribosome, it is actually the cellular machinery. The ribosome is the cellular machinery responsible for synthesizing proteins. So it is the one that reads the mRNA and decode the codons, which should code for a specific amino acid that is supposed to be brought, or that is being brought in by what? By the transfer RNA. So ribosome, it's a cellular machinery that is a manufacturing site. In other ways, we can just say it is manufacturing. We can give it as manufacturing. Can be defined as the manufacturing site for proteins. It is a manufacturing site for proteins. That is one other way, or another way of explaining it, that the ribosome, it is a manufacturing site for proteins, which you can see that we are saying cellular machinery for synthesizing production of proteins. Production of proteins, which I think you can see in this regard. So having said that, let's just quickly move on again. Let's go into the process in detail. I have mentioned and identified all the components that I have been demonstrating and illustrating, actually like identifying on the diagram that I used at first. I have explained all of them. Let me quickly just go into the detail of protein synthesis. it is actually a highly complex procedure that involves a great number of chemical reactions. It is just a complex, as to say, it's more detailed process that has to have a number of chemical reactions, both inside and outside the nucleus, which we say that both inside, I said, when we look into inside, we are talking about the form, we are saying during the formation of what inside, when does it do what, when does it work inside? When does it have to do inside? Inside, it is inside during mRNA, mR during mRNA formation. It is inside during mRNA formation, inside during mRNA formation. I'm sure all this is really making sense to you guys. We are saying both inside and outside, which is outside, outside, outside. We are saying that is during what? Protein synthesis. Outside is actually during protein synthesis. So what I'm saying is a complex of procedures are taking place. These are actually, there is a great number of chemical reactions that are taking place both inside. Inside, we are saying when the mRNA is being formed, and also outside during protein synthesis, which are catalyzed by a host of, we are saying it is being speed up, which are being speed up, being catalyzed, activated, or speeded up. If they are being speed up, catalyzation, it means to say that they are being they are being activated by a host of enzymes. I told last time that there are enzymes that are responsible or chemical messengers or chemical substances that enables to speed up the processes that are taking place. So there are a variety of enzymes that are there. Uh, Translation is the process by which the genetic code carried by the mRNA, the genetic code carried by what? Process by which genetic code that is carried by mRNA is decoded to produce a specific. It is decoded as to say that it's being read by the ribosome. We are saying to is decoded to produce a specific sequence of amino acids, ultimately resulting in a polypeptide chain that falls into a functional protein. I'm sure that from that diagram that I showed first, I'm saying when a series of proteins, amino acids are joined together, they form a peptide bond. The peptide chain is a chain of amino acids, that falls into a functional protein. So that process actually, guys, understand that this occurs in the ribosomes. It occurs in the ribosome, a complex machine in the cell that facilitates the docking of what? Of mRNA. We are saying the attachment of mRNA, which is the ribosome. It allows the attachment of mRNA and the transfer RNA molecule that carry the amino acids to say they do relate, they correspond, or they communicate to each other, which means to say that the ribosome, like I said, that it actually provides the complex function of protein formation as the manufacturing site where proteins are to be formed. Uh, I'm sure we are still together in this regard. So each three nucleotide sequence, which I mentioned firstly there, I said each three nucleotide sequence, which is the codon on the mRNA, corresponds to one amino acid. If you can remember very well, I gave you say examples of amino acids of codons and their corresponding amino acid is specified by a genetic code that it is there. So what we are saying is that the three nucleotide bases or the three nucleotide sequence, which are the codons on the mRNA, they correspond or they code for a specific one amino acid that is specified by the genetic code. I'm sure we are still together, class, in this case. Are we still together here? I'm saying, which again, we can give, for example, the codon AGU. For example, we are talking about an example of the codon as to say that we have got AGU, AUG, codes for the amino acid, the methionine. This is an example to say that you might be given there to say that the codon AUG codes for specific amino acid. Identify the amino acid that it codes for. Normally, this is given. There is a table that is always given there for you to understand and see what is happening. So AUG, adenine, uracil, and guanine, they code for an amino acid that is methionine, and also serves as the start signal of translation. So we are saying when protein synthesis is to start, the AUG, they are also a start codon, which also, when this comes, whenever proteins are to be manufactured, AUG is to start, which is actually initiating the process of protein formation. It is actually signaling or giving a communication that protein synthesis must start. So as the ribosome moves along the mRNA, the ribosome, it moves along the mRNA, tRNA molecule match their complementary anticodons. Are we together in this case? The tRNA, it actually, it matches the complementary anticodons sequence to the mRNA codons. Are we together? I said that we can have, we are given, for example, see, let me say that we can be given, as to say that we can be given, let me give an example that we can be given what, we may be given C, G, and then these are the mRNA. We are saying, for example, these are the codons. These can be the codons that we are given there, as to say these are our what? Our codons. And then you are supposed to identify the corresponding, you must supposed to give the corresponding anticodons to them. The corresponding anticodons to them, we are saying this is to match also to say we have got G, C, as to say these will be the corresponding anticodons it will be the corresponding anticodons to the codons that we are given, which is in response to the question that we are answering here, to respond to the question that we are answering here, to say CCG, the codons, and then the anticodons will be also GC, which also represent to say these are what? They are the anticodons. Just to say they are our anticodons there. You understand? So class, take note of this relationship, codons, anticodons relationship. It is actually a complementary relationship that exists there, which has to do with the matching of the bases in regards to their what? To their normal pairing that we talked about in the beginning. So they bring the appropriate amino acids into place. The amino acids are brought in, appropriate amino acids are brought into place by the what? They are brought into place by the anticodons that are bringing the specific amino acids which are coded by the codons that are on the mRNA molecule that we've been talking about. So the ribosome catalyzes the formation of the peptide bond. The ribosome, it catalyzes, it speeds the formation of the peptide bond, which is the series of the series of amino acids between the amino acids elongating the polypeptide chain. So if there are so many amino acid chains that are joined together, we call that a polypeptide chain, polypeptide bond or polypeptide chain until it reaches a stop codon. A stop codon actually which does not code for any amino acid. A stop codon, it's actually a codon which does not code, it does not represent, which does not code, it does not represent for any amino acid, but it signals the end of the translation. It gives to say that the process must end. it gives to the system that it identifies during the process of the protein synthesis that the protein formation or the chain of the peptide bond must end because we have picked or there is an there is what called a stop codon which does not have a corresponding amino acid that can be transferred to it. So finally, the standard genetic code specifies the basic rules by which the nucleotide sequence of mRNA is translated. The standard genetic code, it specifies the basic rules which by which the nucleotide sequence of mRNA is translated. So the order of the mRNA nucleotides, it is actually the one that defines the type of protein to be formed there. It actually defines the proteins to be formed. So I'm sure that you can still see that the genetic code is redundant. This is one thing that I want you to know. The genetic code is redundant, meaning that most amino acids are encoded by more than one codon. Most amino acids are actually what I'm saying is that most amino acids are are encoded by more than one codon, providing some tolerance against mutation. I'm sure you remember to say a change in the structure of a DNA molecule, which results in the different protein to be formed, is what you call gene mutation. Gene mutation, which sometimes again you remember in your previous lessons or wherever you have read, for those who have read ahead of time, that gene mutation, it is actually one of the sources of or one of the causes of genetic variation anyway, which changes the structure or it changes the type of protein to be formed. So, you need to understand that the genetic code actually, it is actually, it gives the amino acids, it actually understands that there are more than one codon that can code for a variety of amino acids to also understand that if a certain amino acid is given there, it cannot be coded by one codon because it is giving the provision or a chance that mutation cannot randomly take place. Mutation, it would affect the type of protein to be formed, which eventually would cause problems in the functioning of an organism. So this code ensures that genetic information is translated with remarkable precision, is to say with the correct precision into the diverse array of proteins. There are quite a number of proteins that can be formed, which perform various functions within the cell. So the code ensures that the genetic information is translated with a remarkable precision into the diverse array of proteins that perform various functions within the cell. And then I'm sure that you should understand that uh, the cell's protein synthesizing machinery ribosome then translate the codon into what? Into uh, into a string of amino acids that will eventually constitute the protein molecule. So what I want you to understand, class, is that protein synthesis is actually the same as translation, which you should understand that it is according to the codons of the mRNA, which also have to match with the complementary anticodons that are to bring the required amino acid to the ribosome. And then the amino acids are linked together by a peptide bond to form a required protein. And then eventually, in that case, you have already explained enough about protein synthesis. So in whole equation of protein synthesis, if you are to explain it, make sure that there is key terminologies that you should put into your explanation, scientific presentation of your answer. At grade 12, distinctions are possible. It is only because of how you express yourself, understanding the terminology of the subject. So you don't talk about nitrogen bases, what, like that. You need to be specific to say codons are to match to their complementary anticodons bases, which are from the tRNA and the mRNA codons and so forth and so forth, presenting in a very above credit presentation of your answer, which can signifies that you have been in class of life science, you have met or you have gone through Metric Hub. Metric Hub is your best channel for your life sciences, for your geography. All grades are going to be run here. Bring friends and family. We meet again in the next presentation. Just know that 2025 is actually excellent. All things are possible. Distinctions are possible. Grade lower grades are coming. Please link them to Metric Hub. Share to friends, brothers, and sisters. You have a beautiful stay and all the best academic year in 2025. Look forward for the next presentation. Subscribe and turn on your notification button. You have a beautiful stay. And Metric Hub is your best partner for your academic success. Thank you very much.